at the BLS 102, and we are going to look at the topic bacteria. I am just going to run through the topic quickly in preparation for your exam and point out the most important thing. I know some of you might have read this topic before, but there are key things that you should know. It's not everything inside of this thing that you should put in your head. There are key things. Sometimes you might be reading, you think some of those things are important. Those are not the important points. So I will point out the important ones for you. All right. Now, bacteria are single-celled microorganisms. Emphasis on single-celled. You must take cognizance of this single cell. Are you getting it? They are single-cell microorganisms. They are microbe that play a crucial role in various ecosystems and human life. They are among the oldest form of life on Earth, with evidence of their existence dating back to over 3.4 billion years. Now, bacteria are fascinating microorganisms that play an essential role in various ecosystems and human health. Now, one thing you should know is that not all bacteria are bad. Some are good. Those ones are known as normal flora. There are some bacteria that also aid in various metabolic processes in the body. Is that okay? So not all bacteria are actually bad. We have good bacteria and then we have the pathogenic bacteria. The pathogenic ones are the ones that cause diseases. Now, what do we intend to learn in this lecture? We'll focus on the identification of bacteria. How do you know which bacteria is which? The characteristics of the bacteria, their classification, and the ecological adaptation. That is, what these bacteria have that help them to survive in their environment. That is where the ecological adaptation is all about. What helps them to actually survive in their niche, in their environment? What is the functional role of that bacteria in their environment, and how do they survive and adapt to such environment? That is where ecological adaptation is all about. Now, we said bacteria are single-cell microorganisms. I'm saying that again. They belong to the domain bacteria. Are you getting it? They belong to this domain. And there are three basic domains. We have domain archaea, we have domain bacteria, and then we have eukaryotes. Are you getting it? Bacteria, archaea, and eukaryotes. So if they ask you, what are the three domains we have? The three domains, we have bacteria, take note, archaea, that is where you have the algae, blue-green algae and the rest. And then you have the eukaryotes, which are where you find the eukaryotic cell, plant and animals fall under eukaryotes. All right, these are the three basic domains we have. They are prokaryotic organisms. Bacteria are prokaryotic. Now, why do they say they are prokaryotic? Meaning they lack a membrane-bound nucleus or an organelle. Prokaryote, they don't have a true karyote, which is more like a true nucleus. Are you getting it now? And they lack a membrane-bound nucleus and organelles. If you look at the structure of the bacteria, this is the structure of bacteria. You agree with me that you don't see, this is a cell membrane, but the nucleus is not enclosed in a membrane. Like there is nothing actually covering the nucleus. This is the nucleus you can see here. Is in the D. We have the circular DNA. It loops around each other. Are you getting it? But it is not bound by anything. It's just free inside of the bacteria cell. Is that understood? It's just free inside of the bacteria. There's nothing like anything bounding it or covering it, unlike the plant cell, plant and animal cell, where you will see the nucleus inside somewhere like this, and there's something like protective what goes in and out of the nucleus. For that of bacteria, there is no true nucleus, no organelles. Are you getting it? We have few organelles, but they are not membrane bound. That is, something does not surround it. So that is that. These are some of the features of the bacteria. You have the cytoplasm. The cytoplasm is where those organelles are just scattered. That is where the nucleus is also scattered. So in the bacteria cell, the nucleus is in the cytoplasm. Nothing bound it. Are you getting it? This region, it is called the nucleoid. Where you find the DNA is called a nucleoid. Is that okay? Where you find the DNA is called the nucleoid. Why for eukaryotic cell, where you find the DNA is called the nucleus. But for bacteria, the nucleoid is just there. As in, there is nothing. You can see all these wire kind of something here. Are you getting? That is where you find the DNA. Nothing bound it. So you cannot say it has a nucleus. Is that okay? Now, this is the flagellum for movement. This is the cell wall, which we are going to look at later. The cell wall helps us classify bacteria into gram-negative bacteria and gram-positive bacteria. The cell membrane control what goes in and out of the cell. Then we have the pili, and then we have the flagellum for movement, of which we are going to look at each of these things as time progresses. All right. Now, we said bacteria are incredibly diverse and can be found in a wide varieties of environment, including soil, water, and even within the human body. Most times they say microorganisms are ubiquitous. Ubiquitous means that you can find them everywhere. That is the ubiquity of microorganisms. They are found everywhere. Are you getting it? They play a crucial role in various ecological processes, including nutrient cycling, biodegradation, and symbiotic relationship within organisms. You know what symbiotic relationship is now, where two people are benefiting from each other. Some bacteria are pathogenic, like I said. When you say something is pathogenic, it means it has the tendency of causing disease. It is disease causing. Are you getting it? Some bacteria are pathogenic. That means they cause disease. Are you getting it? They cause disease in both humans, animals, and even plants, whilst others are beneficial and used in various biotechnological and medical applications. For instance, your penicillin, the one you take, all these ones, they are from bacteria. Are you getting it? We got you from bacteria penicillin G. Then there are normal flora in your mouth and even in the vagina for ladies. Those normal flora, they are good bacteria. Are you getting it? So there are the good ones, and then we have the ones that are pathogenic. Is that okay? That is that. Now, let us talk about some of the features of the bacteria which we've looked at. We have the cell membrane. We said the cell membrane is a lipid bilayer. So you might be asked, the cell membrane of bacteria is made up of what? Lipid. Are you getting it? It's a semi-permeable membrane. We can say it's a selective barrier regulating what goes in and out of the cell. The cell membrane of bacteria is not more like the cell membrane of plant and animal cell too, which we say regulates what goes in and out of the cell. Are you getting it now? And the cell membrane is also involved in energy production through processes such as cellular respiration and photosynthesis in some bacteria. So the major function of the cell membrane is it regulates what goes in and out of the cell, one. And in bacteria, it's also involved in energy production through processes like cellular respiration and photosynthesis. So you might be asked, what are the functions of the cell membrane in bacteria? Regulate. The major function is that it regulates what goes in and out of the cell. But it also has peripheral functions like cellular respiration and photosynthesis in some bacteria. Now, what about the cell wall? Surrounding the cell membrane. So after you have the cell membrane, but the external part that is surrounding the cell membrane is the cell wall. So first of all, you have the cell wall, then the cell membrane inside. Are you getting it? Which provides structural support and protection. Major function of the cell wall is for structural, more like your wall, structural support and protection. It is composed mainly of peptidoglycan. This is a very important thing you should take note of. Peptidoglycan. The bacterial cell wall is made up of what? Peptidoglycan. It is a combination of what? Protein and carbohydrates. Are you getting it? It's a conjugated protein. Hence, you call it peptidoglycan. That is what the bacterial cell wall is made up of. This is very important. Take note of it. The bacterial cell wall is made up of peptidoglycan. The cell wall maintains the shape of the cell and prevents lysis in hypotonic environment. What is hypotonic environment? Hypotonic environment is an environment whereby the inside of the cell has more concentration than the what? Than the outside. Such an environment is hypotonic to the cell. Inside has higher salt concentration as compared to the what? To the outside. Are you getting it now? Such an environment is hypotonic. And what do you think will happen? If the environment is hypotonic, what do you think will happen? There will be flow into, and before you see it, the cell will absorb water, and before you know it will swell and burst. Are you getting it now? So the cell wall also maintains the shape and prevents the lysis. Lysis means like the breakdown or the bursting of that cell. in hypotonic environment. Now, the composition of the cell wall varies between different bacterial groups. And this is why we classify them into gram-positive bacteria and what? Gram-negative bacteria. So based on the cell wall, bacteria are classified into two. We have the gram-positive bacteria and we have the gram-negative bacteria. The gram-positive, everybody take note of this. The gram-positive, they have what we call the thick layer of this peptidoglycan. Thick layer of peptidoglycan. While gram-negative, they have a thin layer of peptidoglycan. Gram positive, thick layer of peptidoglycan. Gram negative, thin layer of what? Peptidoglycan. Is that okay? So you should take note of that. Is that understood? Now there's something I want to give us. Example of gram-positive bacteria, you have Staphylococcus, the Staphylococcus, Streptococcus, and Bacillus. All these ones, they are example of what? Gram-positive. Staphylococcus, Streptococcus, Bacillus. All those ones, they fall under gram-positive bacteria. All right? Now, how about the gram-negative? Like we said, they have thin peptidoglycan, but they contain lipopolysaccharide. They contain what? Lipopolysaccharide instead. Lipopolysaccharide. All right? Instead. All right? And example is E. coli, Salmonella, and Neisseria. E. coli, Escherichia coli, Salmonella, the one that causes typhoid, Salmonella typhi, are what? Neisseria. Neisseria is the one that causes what? Gonorrhea. Neisseria gonorrhea. All those ones, they fall under the gram-negative bacteria. So I said we have gram-positive bacteria. Then we have what? Gram-negative bacteria. Is that okay? Exactly. So we have gram-positive, gram-negative. Say gram-positive, they have thick layer of peptidoglycan. Gram-negative, they have thin layer of peptidoglycan. Now, there's a staining technique that is actually used, of which we are going to discuss later. They usually stain them with a purple stain. And for the gram-positive bacteria, they retain the purple stain. Are you getting it? They retain the purple stain. Why for gram-negative bacteria, see that you call them purple stain or you crystal violet, more like the color of this marker or this write-up. Why for the gram-negative bacteria, they stain pink instead of purple. Is that okay? They stain pink instead of what? Purple. You should take note. They don't retain the violet color, but they stain either pink or red. So that is the difference between gram-positive and gram-negative bacteria. Like I said, gram-positive has a thick layer of the peptidoglycan. They don't have a lipopolysaccharide. Are you getting it now? They stain purple or crystal violet. Example are Streptococcus, Staphylococcus. Are you getting it? All those ones, they are, and Bacillus, the example of gram-positive bacteria. While gram-negative bacteria are the ones that have thin layer of peptidoglycan, but comprises of lipopolysaccharide. Example is E. coli, Salmonella, and what? Neisseria. E. coli, Salmonella is the one that causes typhi, Salmonella typhi. Then Neisseria is the one that causes what? Neisseria gonorrhea. All right. So that is that for the composition. Emphasis on the bacterial cell was composed of peptidoglycan. Now, the cytoplasm. Inside the cell, the cytoplasm is a gel-like substance that houses various cellular components. It contains essential molecules such as enzyme, ribosomes, and nucleic acid. That is for the cytoplasm. All right. Now, what about the ribosome? The ribosome is still the same thing with the ribosome for normal cell. in that they are responsible for protein synthesis and they are abundant in the cytoplasm and they play a role in translating genetic information into functional protein that facilitates various metabolic processes. Like we said, we have DNA to what? RNA to what? To protein. Where we said that DNA to RNA is transcription, then RNA to protein is what? Translation. All right? Exactly. So they are responsible for translating genetic information into what? Protein. Responsible particularly for protein synthesis. That is the ribosomes of this is more like the normal ribosomes we'll be talking about. All right. Now the nucleoid. Like I told you earlier, that the bacterial cell do not have a true nucleus, but they have circular strand of DNA. And these circular strands of DNA, now this one when they see when they fold here, are you getting it? They are actually found in a region called the nucleoid. So what you have here is the nucleoid. All right. This region that contains this circular strand of DNA is the nucleoid. It does not have a membrane-bound nucleus. The difference between bacteria and the eukaryote is that they don't have that membrane-bound nucleus like those of plant and animal cell, the eukaryote. They are just free. The DNA, they are circular in nature and they just round up like that. Are you getting it? The nucleoid contain genes that encode proteins crucial for the cell survival and reproduction. It is the nucleus that have the DNA, which will later be converted to what? Protein through that processes I listed earlier, DNA, RNA, protein. Is that okay? Take note, they don't have a true nucleus, but they have a nucleoid, a region called nucleoid. Emphasis on this also. Now, flagella or pili. Now, in addition to this major component, many bacteria possesses flagella or pili. Flagella are long whip-like structure that enable bacterial motility. So the use of the flagella is for what? Motility, allowing them to swim towards nutrient or away from harmful substance. Pili, on the other hand, are shorter hair-like projection that facilitate adherence to surfaces. So when the bacteria wan hook for somewhere, na the pili dey use. Why if it wants to move from one place to another, it uses the what? The flagella, singular flagellum. Now also, the pili also play a role in the transfer of genetic material between bacteria through a process known as conjugation. So genetic material can also be transferred through conjugation using the what? Using the pili. So pili has two functions. One, for adherence, to attach to surfaces. When the bacteria get to where they go, if you want stay there, na the pili you go use. Also, it can also be used to transfer genetic information between bacteria through a process called what? Conjugation. Are you getting it now? So the process of conjugation in bacteria happen as a result of what? The pili. Presence of pili there. So pili has two functions: adherence and what? Transfer of genetic information. Is that okay? So take note of that. Now, identification of bacteria. How do we identify bacteria? Now, identifying bacteria species is crucial for understanding their role in health. For instance, how do you know that this bacteria is this so bacteria that is present and you're identifying them? Are you getting it? A group of bacteria that can be seen with the naked eye is called a colony. Please take note. A group of bacteria that can be seen with the naked eye is called what? A colony of bacteria. So how do we identify these bacteria? Are you getting it? One of the methods is morphological identification. Morphology has to do with how the bacteria look, the form. Another method is by staining techniques. Another method is by biochemical techniques. Another method is molecular techniques. Take note of these four. Emphasis. Morphological identification, how the bacteria look physically. Staining techniques, that is where you use gram-positive and gram-negative. You stain the bacteria to tell you whether the bacteria is a gram-negative bacteria or a gram-positive bacteria. Then use biochemical test. These biochemical tests, there are some certain bacteria that they produce certain substances. Those substances are used to identify them. Are you getting it? There are some that when you place them with food, they will ferment the food. Some of them are called lactose fermenters. There are some that are aerobic. That means they produce oxygen, they utilize oxygen. Some are anaerobic, they don't utilize oxygen. Are you getting it? All those ones, they fall under biochemical tests. Then molecular techniques like PCR, polymerase chain reaction, and so on, are also used to identify bacteria. Now, morphological characteristics. We say this method examines the shape, the size, and arrangement of the bacterial cell. And common shapes are cocci. Cocci are spherical in shape, bacillar are rod shape, and spirilla are spiral shape. Now, the one that they may ask you is, cocci, as the name implies, cocci, spherical. Bacilli is rod shape. A rod shaped bacteria is called bacilli. Emphasis on this one. Take note. Spirilla, you can always remember it from spiral shape. Spirilla. Cocci, you can always remember that this one is spherical. But bacilli, rod shape. Emphasis on bacilli is what? Rod shape. So they may say cocci, bacilli, spirilla is what? Under morphological. This one is talking about the shape of the bacteria. These three, bacilli, cocci, based on shape, bacteria can be classified as cocci, bacillus, spirilla. Are you getting it? And even vibro. Vibro is comma shape. Are you getting it? Vibro is what? Comma shape. Emphasis on this. They can also be classified as single, pairs, chain, or cluster. That is where you have something like staphylococcus, streptococcus. Are you getting it? Chain. You can classify them like that. Now, on the staining techniques, I have explained this before. Emphasis on the person that actually does the staining or that developed the staining is Hans Christian Graham in 1800. Are you getting it? Hans Christian Graham in 1880s. Now, this method differentiates bacteria into gram-positive bacteria, which retain the crystal violent stain, and gram-negative bacteria, which do not, based on the structure of their cell wall. Like we said, that the ones that have thick layer of peptidoglycan, that is gram-positive bacteria, will retain the crystal violent color, while the ones that have thin layer of peptidoglycan and lipopolysaccharide, the gram-negative bacteria, will not take in that color, but rather they will stain pink in color. Is that okay? He said this distinction provide critical information about the bacterial structural characteristics and inform treatment options, as gram-negative bacteria are typically more resistant to antibiotics. Gram-negative are more resistant to antibiotics. Why are they more resistant? Because most of the antibiotics, they are developed to work on the cell wall, to destroy the structural integrity of the cell wall. So if most antibiotics are made to actually destroy structural integrity of the cell wall, a bacteria that normally does not have a cell wall, it has other coping mechanism. Are you getting it? So it will be very, very difficult to target to kill that bacteria, since most antibiotics work by cell wall destruction. Are you getting it now? That is why we say gram are typically more resistant. Why? Because they no get cell wall when we say their cell wall does not contain that peptidoglycan that you can easily destroy and kill the bacteria. They don't have it. Are you getting it now? So it's very difficult for you, and hence they will resist antibiotics. What antibiotics? They are drugs that actually kill this bacteria. They can kill, that is bactericidal. They can just stop the bacteria from multiplying. That is what? Bacteriostatic. We have two: bactericidal, antibacterial agent, bactericidal, and then we have bacteriostatic. This one kill the organism. Why bacteriostatic? They don't kill the organism, they just prevent the organism from multiplying and from increasing. So we say the gram stain, a differentiated staining technique, categorize bacteria into two groups. We have the gram positive, which retain the crystal violent color, and the gram negative, which do not. This one stain pink color. All right. So that is that. Now, there are biochemical tests that you can actually use to determine the kind of bacteria that is present. Is that okay? There are various biochemical tests that can be used. These biochemical tests, they depend on the ability of these bacteria to ferment sugars. We have lactose fermenters and some that are not lactose fermenters. Are you getting it? Some bacteria, they can ferment lactose. What is lactose fermenters? Like, they convert lactose to what? To alcohol. and carbon dioxide. Are you getting it? Sugar to carbon dioxide. Those ones you call them sugar fermenters. Lactose fermenters are not lactose fermenters. Lactose is a sugar now. Exactly. Now, some of them produce gas, or some of them they act on specific substrate, like they act on specific things. Now, for instance, catalyst test distinguish between catalyst positive, that is aerobic, and catalyst negative, anaerobic bacteria. Are you getting it? The catalyst test help to distinguish between catalyst positive, that means aerobic bacteria, the one that use oxygen, and catalyst negative, anaerobic, the ones that do not use oxygen, based on the ability to break down hydrogen peroxide. Now, this catalyst test, it works based on some organisms. When you see them, they are able to break hydrogen peroxide into what? Into H2O plus what? O2 gas. Then when you balance the reaction, you will be able to, when you balance the reaction rather, at least you'll be able to distinguish. Are you getting it? So that is that. Now at the end of the day, of course, here will be 2, and then we can have here as 2, and this equation is balanced. Example of catalyst positive aerobic organism, you have staphylococci. Staph is catalyst positive, while strep is catalyst negative. Is that okay? Staph catalyst positive, strep catalyst negative. Is that okay? Now, this is based on their ability to break down hydrogen peroxide. Please take note, this question might come out. Example of catalyst positive organism, staph. Catalyst negative, strep. And the test, it works by ability. So there are certain organisms, what you do is just to get the hydrogen peroxide, put it here, and then put the organism. Let us just say you put the organism at the end of the day. Now, at the end of the day, you are going to see a gaseous film, like gas coming out. Are you getting it? By so doing, you know that that particular organism is what? Catalyst positive. Is that okay? And is tendency of being what? A staphylococcus. Then if it does not have that ability to break down that hydrogen peroxide, it means it is what? Anaerobic, which is catalyst negative. So the catalyst test help to distinguish between aerobic and anaerobic bacteria. Please take note. The catalyst test help to distinguish between aerobic and what? Anaerobic bacteria. Is that okay? Now, we said this test assess the metabolic capabilities of bacteria, such as the ability to ferment sugar or produce specific enzyme. Example include catalyst test, oxidase test, and urease test. All these are some biochemical tests: catalyst test, oxidase test, urease test, and so on and so forth. Oxidase test, the ability to oxidize something. Urease, ability to what? Urease test, to break down uric acid, something like that. Think so. Urease test. I learned this in year 2 pharmacy. That can, yes. The urease test is used to identify bacteria that can produce the enzyme urease, which hydrolyze urea into ammonia and carbon dioxide. Is that okay? So that is that. Example of urease positive, we have Helicobacter pylori, Klebsiella species, Proteus species, but nobody will ask you those examples. So just know we have tests such as catalyst test, oxidase test, and urease test. Each of these tests, they just more like if you put the bacteria and some of these substances there, what will happen? If it gives you a positive test, it means, oh, that particular bacteria is catalyst, is urease positive, and so on and so forth. Alright? So if you have urea here, and then that urea, you put a particular bacteria here close to the urea, and it breaks that urea into ammonia and oxygen, you know that that bacteria is what? Urease positive. Is that okay? Urease is an enzyme. So like we said, the biochemical test, it makes use of the ability of these organisms to actually do what? To produce certain enzymes or to produce gases, or to ferment sugars. Is that okay? So let me run to the next one. Other molecular techniques. Now, the advent of molecular biology revolutionized bacterial identification. Techniques such as polymerase chain reaction. Polymerase chain reaction is more like a replication technique where you amplify a particular gene of interest. Ribosomal RNA sequencing and whole genome sequencing allow for precise identification of genetic material. Advancement in molecular biology have introduced techniques such as polymerase chain reaction and DNA sequencing for bacterial identification. So molecular techniques that are used, we have polymerase chain reaction, DNA sequencing, ribosomal RNA sequencing, and whole genome sequencing. This method allow for detection of specific genetic markers, facilitating the identification of even closely related species or novel species. And I don't think anybody will give you deep questions under these molecular techniques because it's actually beyond your scope for now. But just know the various techniques. Polymerase chain reaction is under molecular technique, ribosomal RNA sequencing, whole genome sequencing, and then we have DNA sequencing. All right. Now, bacteria character. Let's look at some of the characteristics of bacteria briefly. We say bacteria ranging in size from 0.2 to 10 micrometer. They come in various shapes, including cocci, bacillus, spirilla, vibro. I've explained this thing before. I say bacillus is rod shape, cocci is spherical, spirilla is spiral, vibro is comma shape. We have explained all this before. Now, the cell structure. We say bacteria are prokaryotic, meaning they lack a defined nucleus and membrane-bound organelle, which we have said. Their genetic material, usually a single circular chromosome, is located in a region called the nucleoid. In a region called the word nucleoid. That means their DNA is found in their nucleoid. Additionally, many bacteria have plasmids are small circular DNA molecule that can carry gene for antibiotic resistance or virulence factor. Please take note. This is very important. What are plasmids? They are small circular DNA molecule that can carry genes for antibiotic resistance or virulence factor. They carry the gene present there so that in case, say, a particular antibiotic is administered, you know why you take antibiotics so that you can kill bacteria. So they also have something inside of them that can make them resist the effect of that antibiotic, and that is the plasmid. Is that okay? Or any virulence factor, like what could aid them to cause their harm. They are stored in the what? In the plasmid. And plasmid is a small circular DNA molecule. All right. Now, the cell wall composition. The majority of bacteria possess a rigid cell wall composed of peptidoglycan. We've talked about that. A polymer made up of carbohydrates and protein, that is sugars and amino acid. We've said all that, where we said the cell wall is what causes the staining process, where we have the gram-positive bacteria and then we have the gram-negative. Now, classification. Of course, we said that bacteria is found under the domain bacteria. There are three other domains. The three domains are bacteria, archaea, and eukarya. Now, the phylum, we have other phyla in the bacteria called the proteobacteria, Firmicutes, actinobacteria, and cyanobacteria. Each of these, they have their distinct class, other families, and so on and so forth. Just talking about taxonomic classification. So that is that. And the species, they are very much so, due to the complex nature, there is very difficult to define their species. All right. And I believe that is everything. Another thing again. Please take note of this. The concept of operational taxonomic unit based on sequencing data is often used in environmental microbiology to categorize bacterial diversity. Take note of this statement here. All right. Now, ecological adaptation of bacteria. How do bacteria stay in the body or in their environment without being dead? Now, bacteria exhibit a remarkable ecological adaptation, allowing them to thrive in diverse environments from extreme conditions to the human body. Bacteria are extremophiles. What do you mean by extremophile? They stay in very extreme conditions. Places that normally you don't even see normal, you find bacteria there, especially the archaea. Are you getting it? You find them there. Now, the adaptability is attributed to several factors. Environmental resilience. We say many bacteria are extremophiles, a very extreme condition. Places that you never want to see life there, you see bacteria. And archaea too, you also find them in places that archaea, is another domain of its own, right? So, but you see bacteria in very extreme places, like very hot temperature places, places where pH is very either high or low, places where the salt content is very high, you will see bacteria there. For instance, thermophiles. Thermophiles are bacteria that thrive in at high temperature. Take note, bacteria that thrive at high temperature, they are called thermophiles. Halophiles are bacteria that flourish in high salinity environment, that is high salt environment. Huh? High salt environment, you find the halophiles. Are you getting it? These adaptations enable bacteria to colonize niches that are inhospitable to most form of life. What do we mean by that statement? Because of all these things, bacteria, any place where we say you not normally other living organisms of its day, you go see bacteria there. We say thermophiles, as the name implies, thermo-loving philes, high temperature region. Halogen, salt. Are you getting it? They flourish in very high salt or salinity environment. Then another thing that makes bacteria to be able to adapt to their environment is that they form biofilms. Biofilm is just like an enclosed case where the bacteria can hibernate inside. Let us say this is the bacteria cell. They can form that biofilm, they are polysaccharide and polymeric substances. Are you getting it? And that biofilm will just cover that bacteria. The bacteria is inside though, so anything when they do, the biofilm cover them. Are you getting it? It will not affect the bacteria. Is that okay? Now, we say bacteria can form biofilm, aggregate of microorganisms that adhere to surfaces and encase themselves in a protective matrix of extracellular polymeric substances. Different bacterial microorganisms, you see them, but they are enclosed by a protective matrix of extracellular polymeric substances. Now, biofilm enhances bacterial survival in challenging environment by providing protection against desiccation, antibiotics, and immune response. The biofilm is just be like something when we say they cover the bacteria there. Made up of what? Extracellular polymeric substances. Is that okay? So that is everything you need to know about bacteria. Questions under the bacteria. Now you have the first question, yes, which says, what is the domain of the bacteria? Of course, we said the domain of the bacteria is the bacteria. There's nothing there. Which structure is bacteria regulate the entry or exit of substance? That is the cell membrane. What is the main component of the bacterial cell wall? We said the main component of bacterial cell wall is peptidoglycan. While gram-negative, they have lipopolysaccharides. Which bacterial structure is responsible for its motility? The flagella. All right. Then let's go to the next one. The next question says, the nucleoid region in bacteria contains the DNA. DNA in the bacteria is stored in the nucleoid region. Bacteria reproduce through binary fission. That is how they reproduce. Which term describes bacteria with a spherical shape? We say cocci, spherical. Rod shape, bacilli. Which staining technique distinguish gram-positive and gram-negative bacteria? Is the Gram stain by Christian Ham Grams. Is that okay? Then we have the next one. Gram-negative bacteria appear what color? Pink or red. They don't appear purple. Which of the following is a molecular method of identifying bacteria? We say we have gram stain. Molecular is PCR, polymerase chain reaction. What bacterial structure is involved in conjugation? Like we said, that bacteria can actually transfer genetic information via what is it called? They can transfer genetic information via the pili. Are you getting it? In a process called what? Conjugation. So the correct answer is pili. Which of the following is not a shape of bacteria? Cuboidal. We have spirilla, cocci, bacilli, vibro, but there's nothing like what? Cuboidal. So please take note of that. Then the next question. What is the role of ribosomes in bacteria? Ribosomes in bacteria for what? Synthesis of protein. A rod-shaped bacterium is called, of course, a rod-shaped bacterium is called bacillus. What is the function of the bacterial plasmid? We say bacterial plasmid carry extragenetic traits like antibiotic resistance and also virulence factor. All right. So option D. Which test detects the presence of catalase enzymes in bacteria? It may not come as catalase enzyme. They will ask which test actually differentiate between aerobic bacteria and what? Anaerobic bacteria. The correct answer is what? Is catalase test. All right. Urease test the differentiate between urease positive and what? Urease negative. All right. Then the next one. In which region is bacterial DNA found? Bacterial DNA is found in the nucleus. What type of bacteria thrive in salty environment? We say halophiles. Are you getting it? Very thermophiles is very high pH. Acidophile, very acidic environment. All right. The primary function of biofilm is, like I told you, bacteria form biofilm for what? For bacteria form biofilm using what? Extra polymeric substances. Option C. All right. Then we have the next one. The gram stain technique was developed by who? Hans Christian Grams. Option C. Bacteria that require oxygens are aerobic. Are you getting it? What is the smallest taxonomic unit in bacterial classification? Of course, it's the species. Which of the following is not a biochemical test? PCR is a molecular method. Polymerase reaction is not a biochemical test. All right. Then we have the next one. What allows bacteria to survive in extreme heat? Thermophilic enzymes. Are you getting it? What is the typical size range of a bacteria? It's 0.2 to 10 micrometer. 0.2 to what? 10 micrometer, typical size. The bacterial cell wall helps prevent osmotic lysis. Like we said, that if it is placed in hypotonic environment, right, the cell will help prevent osmotic lysis. What allows bacteria to attach to surfaces? We said is the word pili. That's what allows bacteria to attach to surfaces. All right. The next one. What kind of genetic material do bacteria typically have? They have circular DNA in a nucleoid. Option B. Then the domain bacteria is separate from archaea and eukarya. Right? Which method provides the most precise bacterial identification? Is it gram stain, morphological, biochemical test, and DNA sequencing? The most precise is DNA sequencing, which is a molecular technique. A common shape bacterium is referred to as what? Vibrio cholerae, the one that causes what? Cholera. All right, is a vibrio shaped bacteria. And then we have the last one here. Bacterial conjugation involves transfer of DNA through the pili. Which group of bacteria has thick peptidoglycan layer, gram positive? What is an extremophile? A bacteria that lives inside human, no. A bacteria that causes disease, no. A bacteria that thrives in extreme condition, inhospitable condition. Place where you say you no suppose see anything, you dey see them there. Are you getting it? So that is that under bacteria. I believe with this, you should be able to do very well in bacteria. Take note of the key points we looked at. Are you getting it now? The key points.